Hey you guys, what's up? Welcome to science class. Did you know that cells require precise spatial organisation of their cellular components in order to function efficiently? How is this achieved, you ask? Well, one key mechanism that is implicit is RNA localization. RNA localization is a fascinating process by which mRNAs are transported and anchored at precise cellular locations. These processes occur in numerous cell types and organisms during various biological processes and plays a vital role in protein synthesis. Do you want to know how RNA localization works? Let me tell you. The process can be divided into several distinct steps. First, the mRNA is processed in the nucleus. During the process, the cis-acting nucleotide sequence elements, known as zip codes, are recognized by RNA binding proteins which mark the RNA for localization. Second, following export from the nucleus, a fully transportable component is formed called the ribonucleoprotein particle, or RNP. Third, the transport RNP associates with molecular molecules such as kinesins and dyneins and are transported along the cytoskeleton. Finally, the RNA must be anchored at the final destination. There must be a mechanism to release the mRNA from the transport anchoring complex to allow the mRNA to translate it at the final destination.
There have been a multitude of discoveries in the mechanisms of RNA localization, and over the past decades of research, one certain mechanism has been found to have a major influence, which are microtubules. One of the most significant discoveries in regards to the mechanisms of RNA transport along microtubules is its polarity behavior in order to direct macromolecules across the cell. One study created a method to localize RNA within two differing polarized mouse cells, being a neuron and the second an epithelial cell that lines the intestine of mice. The results showed that molecules were directed towards the bottom pole of the epithelial cell, and the RNAs traveled towards the extremity of the growing plus end of microtubules. This discovery disproved the typical deception of a self-targeting behavior. This new insight into RNA gravitating to the polar ends of microtubules within cells has been utilized to predict where RNA can end up and further development of artificial mRNA and their targeting. The cooperation of motor proteins in the mechanisms of RNA localization is still unclear. Though some models have been proposed, such as kinesin indirectly activating dynein, or that the two proteins may use each other for travel, with kinesin attaching to dynein and vice versa.
The reason mRNA localization is so advantageous is firstly because it produces proteins at their site of action, which prevents the cell from having to regulate the protein activity before they reach their target actions. Secondly, localizing mRNAs allows the cell to regulate the expression of certain genes independently in different regions and to rapidly generate proteins whenever needed. Thus, local translation of proteins affords more spatial control and is faster and more economical for the cell.
An example of cellular organization is evident when specific mRNAs are directed to one daughter cell via RNA localization. This is seen during mouse embryogenesis. A specific mRNA called CDX2 mRNA and its localization were shown to play a role in determining trophectoderm fate. When CDX2 levels are higher in a cell, it's more likely to become part of the trophectoderm. In general, when specific mRNAs are localized in a cell during embryogenesis, they ensure that daughter cells inherit distinct molecular components. So, in this way, we see that RNA localization is crucial in cellular organization to such a degree that it determines not only organization, but also development and specialization. We said before that RNA localization is implicit in efficient cell function. But what exactly does that entail? When we say efficient cell function, we're referring to the ability of a cell to carry out its biological processes in a way that maximizes its use of resources, minimizes the waste, and achieves its intended goals with minimal energy expenditure. RNA localization aids in this efficiency because a single localized RNA molecule can lead to localization of multiple copies of a protein in a target compartment, without the need to transport each protein individually. Cell cycle control refers to the regulatory mechanisms of the processes that lead to cell division. The specific mRNAs that code for cell cycle regulatory proteins are localized to subcellular locations. For example, cyclins and cyclin-dependent kinases are proteins that play a role in cell cycle progression. The mRNAs that encode them can be localized to the nucleus or the cytoplasm, which ensures that these proteins are available when and where they are needed for cell cycle transitions. Through this, we can see how RNA localization plays a key role in cellular organization and function.
The incorrect localization of RNA caused by defective RBPs has been correlated with many kinds of non-infectious diseases, such as neuromuscular diseases, cancer, and developmental disorders. In a study on ASL, a disease characterized by a loss in motor neurons, its distinguishing mutations were found to affect RNA metabolism, resulting in alternative splicing, destabilization, and incorrect localization. Particularly, it was found to affect the RNA binding protein TDP43. Similarly, in SMA, it was found that insufficient production of the RNA binding protein SNM resulted in the insufficient localization of the mRNA transcripts for acetylin B and neurotin, resulting in the symptoms of SMA. Various studies of cancer have also shown that RNA binding proteins may be involved in the initiation of cancer. By knocking down the non-coding RNA Samson, melanoma viability was found to increase. Furthermore, the overexpression of the gene GERNA was found to disrupt the BRCA1 protein, reducing its ability to suppress tumours. RNA localization has also been correlated with developmental disorders. In the stem cell disease dyskeratosis congenita, incorrect localization of the TERT mRNA was found to induce telomerase shortening. By better understanding the mechanisms of RNA transport, we can create more effective therapies to treat such disorders. Thanks for watching today's video. As a quick recap, today we learned what RNA localization is, the mechanisms of localization, the importance of localization, and its clinical relevance.
Thanks for watching!